Good morning, everybody. Lovely to be back. And so, if everybody can hear me, I'll get started. And also, I want to say that today's topic would be a first time for me too. So, it's a very complex topic. So, I kind of dwindled it down to be more fun than being complex. So if I do make some theoretical, technical bloopers, I take all the blame because the more important thing is we learn, we are curious. So one more thing I would like to say is, yes, we have the cat around for you to ask questions. That's great. But also, as I'm going through the slides, if there is something that I'm making a little bit complicated, or I introduce a complex term, right? Suddenly I say quantum tunneling and I don't explain myself well. The chat box, I've kept it active. Feel free to just drop a question then and there. I'm one vision of mine will be on the chat box. So that way, we can make the uh, conversation, interaction, everything more interactive. So that is what it is. Okay. So feel free to ask me, interrupt me, or whatever with a question you might have. Okay. So once again, great. This is my second time being around here. Last time I told the story about an organism called agrobacterium. I'm really gratified to see the response I received. So I thought, let me have some more fun. Let me dig into a topic which I am learning myself. So I have this premise that the moment you teach, or the moment you start teaching, you learn better. So that's what I'm doing right now. So I go to a topic where I want to learn more. So what I do is I talk about it. So yeah, thank you, Phil. Yes, I teach, therefore I learn. So that's what it is. All right. So today's topic is about okay, and quantum mechanics has been a very, we journeyed together. My dad is a physicist. Uh, so he says that he used to hold a small, his own science circle at home with his colleagues, and this was 1970s. I was hardly 2 or 3 years. So the first technical word I picked up, he says, was quantum mechanics. Maybe I like the sound. So from then on till here, it's been a great journey. But this is one topic I really love. Okay, so today we'll look at quantum biology, how that quantum mechanics actually came into the life sciences, right? So the robin's flight, the smell of a rose, and the oxygen around us, everything. We science starts with an observation. So one of the greatest observations that was made when quantum biology became a thing was the flight of a robin, and we'll start with that, and then something like the smell of a rose. And of course, we breathe oxygen. Where is that coming from? We thought we had the answers just with life sciences and chemistry, and a little bit of physics, but where did quantum mechanics come into the picture? So let's get started on a great journey. So let's meet our friend, the European robin. Okay. What it does is, it's all of 19 grams in weight. Very, very small bird, a very beautiful bird, I should say, very cute. But it does something amazing. It migrates from one place to the other. The first time they tracked it, it was traveling from Germany to UK. They even tracked it with a nanotag. And it flew for 4 hours to cross that North Sea between Netherlands and UK. Just a 19-gram bird which undertook a journey of 140 miles, meaning that it was traveling at an average speed of 35 miles per hour. Okay, so that's how it is traveling. What kind of a natural GPS does it have? How does it know when to start and when to go, right? That's what intrigued scientists. So everything, as they say, starts with an observation. So But it's not about this one particular journey which I am showing in this flight. The Robin's Odyssey, as we can say, sometimes flummoxes even the greatest ornithologists. So there you have the European robin. It's called Erythacus rubicula, but that's what it is. Yes, has somebody here saying hummingbirds migrate across the Gulf of Mexico, Mexico to Texas, and they are only 2 grams when they arrive. They start at 10 grams. Yes, let me tell you something about the European Robin's Odyssey. So what does it do? Why does it even migrate, right? It starts its journey somewhere in the August of every year. August would be the time when it is in central Sweden, that is in a spruce forest, that's what they called it. You are looking at August, and in the life of a European robin, it just reared some chicks a few months ago. Now suddenly in August, it starts eating. It starts eating double the number of worms, double the number of insects. It starts eating ravenously to become fat. Now, it needs to be free until next spring, and now it needs to escape the winter. So now it is headed south. So it is going all the way from Sweden to the Mediterranean coast of North Africa. Imagine that. If you take a to and fro, it's about 5,000 miles. Now, when it goes, it's precise. It inclines itself to 195 degrees. It has some kind of compass within it, 15 degrees west due to south. It has to pass through towns, lakes, valleys, towns, all kinds of landscapes. 200 miles per day is when it will travel, a little bit of rest, and it will take off again. The destination, as I told you, is from Sweden to the Mediterranean coast, North African coast. What they found was it makes use of a phenomenon called magnetoreception, meaning in the brain of that bird there are certain compasses, and that is a set of proteins. We call them the cryptochrome proteins, as the name says, chrome, something to do with color. So the cryptochrome proteins are located in the retinal cells of the bird's eye. When they dug deeper, what they found was, and let me throw this whole thing out at you without explaining any basics, let's first marvel at what the robin does, and we'll keep coming back to this little bird back and forth as I progress in the talk. So within the bird's eye, there is a cryptochrome protein, and the two most important elements or molecules, one is called the FAD, the flavin adenine dinucleotide molecule. Now, what is this flavin adenine dinucleotide molecule? It's actually is synthesized from vitamin B2, and FAD is involved in cellular metabolism, and basically without FAD, nothing in terms of energy will be reared in any organism. So that is one molecule it uses. The other thing it uses is what is called a tryptophan. Now, tryptophan is an amino acid which makes 3 kinds of compounds. The most important thing is serotonin. Serotonin is a neurological neurotransmitter which is involved in mood swings. So that's one thing that the tryptophan does, right? The other thing is melatonin. Melatonin is what governs the sleep-wake cycles.
Welcome to the Science Circle.
And the third thing is niacin. Niacin is vitamin B3. It is what is involved in energy, digestion, and tissue repair. Now, so these two molecules, FAD and tryptophan, is what constitutes that entity called the cryptochrome. So now, when the robin is flying, it is making use of the Earth's magnetic field, and somehow there is a connection that is built between the Earth's magnetic field and this cryptochrome. The spooky part of it is this. This particular magnetic field we are talking about, the Earth's magnetic field, it's about what you could say is 130 to 70 microtesla is what they say. Now, just to give you an idea of what 30 to 70 microtesla, it is sufficient to deflect a compass needle. But if you look at it, it is 1/100th the magnetism of a fridge magnet, like the refrigerator magnets you put. 30 to 70 microtesla is like 1/100th of that. Now, what is it that it can interact with such a weak magnet? Because that particular 30 to 70 microtesla is just 1/1000th the energy needed to break a chemical bond. So what are we talking about? How come? And if you look at it this way, your magnetic compass can distinguish north and south poles, but the Robin technically can distinguish only between the pole and the equator. So what is happening here? What's happening here? What's happening is when this Robin is flying, there is a photon. Photon is a small blue light, right? That hits the cryptochrome, right? So when it hits the cryptochrome, there is a small electron which jumps from tryptophan to Fad. Now, going back to your what do you call the atomic structure, you know that in the nucleus you have protons and neutrons, and then there is this electron. Electron is the thing which jumps from one element to the other. So when this photon of blue light hits the cryptochrome, a small electron jumps from the tryptophan onto the Fad. The resulting molecules will now have odd number of electrons. Meaning, to be stable, the outermost orbit of an atom should have 8 electrons. Suddenly you have this extra electron, so this odd number of electrons. So they are vying to be paired somewhere. So that's why you call this Fad and cryptochrome, as soon as a photon hits it, they become a radical pair, and we get to what is called the singlet state. Meaning these electrons are now spinning in the opposite direction. Now, spin is a very tricky term. Spin is not as if spinning around an axis, but in case of an electron, what you call spin is simply the direction in which that electron is oriented. So let's, without making it complex, think of a positive spin in one direction and a negative spin in the other direction, right? So when the photon of light hits that protochrome, those electrons oscillate rapidly back and forth and get into what is called a singlet state. At this singlet state, when the electrons are vying to be paired, they get paired with the weak magnetic field of the Earth. Now, when it interacts with that Earth's magnetic field, now they get into what is called the triplet state, when both the electrons now spin in the same direction. Imagine this, thousands, billions of miles and such a weak magnetic field, but this interaction is going on in the bird's brain. So at this state, both states can undergo chemical reactions and they get into what is called the signaling state. And if you can look at that cryptochrome, that Fad molecule and tryptophan inside oval, it also has a tail. The tail gets touching that oval thing. So that's like a charge which goes back to activate that molecule. When that happens, there is a signaling that becomes a signal molecule. The cryptochrome turns on a biochemical cascade that will release neurotransmitter molecule. And as I told you, tryptophan is involved in all that serotonin, melatonin, right? Those neurotransmitter molecule hit the retina. The signals continue to the bird's brain. The magnetic information is integrated with the information with a directional cue, and the bird is exactly getting to the destination, remembering every step of the way, so it can make that travel forward and backward at an exact angle. And this cryptochrome keeps returning to the ground state and the process starting again. So it's a clicking that happens between the Earth's magnetic field and this cryptochrome, and the bird is able to travel 2,000 miles without missing a beat. Most of its flight is at the night time, right? So apart from European robin, they found this flawless magnetoreception even in the bar-tailed godwit, which I am showing you there. So let's just look at this amazing phenomenon. What exactly is going on here, right? So now This is what we do in science. We observe something and then we dig deeper. We want to see whether there is something which is happening in our real life and kind of relate that, right? So yeah, the one of you made a comment, magnetoreception will not work at night. Then no, but the signals have already been triggered. So at night, it is using the FAD tryptophan interactions to continue the flight, right? And yeah, do reptiles have this receptor? This receptor is, as I continue in my talk, you will see that it's not just reptiles, it's in butterflies, it's in fruit flies, whatnot. And yeah, moonlight also has some photon packets. And that's all about this quantum world. You are actually looking at the smallest signals, which can, it's almost like identical twins separated by millions of miles, but they are known to have some signals which trigger each other. That kind of spooky connection between one electron, which is now lying right next to each other, even if they're separated by trillions and trillions of miles, they somehow maintain that connection. That's exactly what is going on. And that spookiness has spooked even the great mind called Albert Einstein. More of that in a little bit. So where do we see this quantum reception in real life? DVDs, your laptop, your cell phone, right? What happens when you get into an MRI detector? All that is happening is this quantum entanglement, as you call it. All it needs is the smallest element in the periodic table, which is hydrogen. What happens when you get into that MRI machine is there is a magnet in that magnetic resonance, as they call it, right? It is simply collecting all the protons inside you, the hydrogen atoms, and which were going around in what they call a drunken walk fashion, like everywhere. It puts them in a military walk. All the hydrogens, due to that magnet, are temporarily walking like an army parade. And that's when those signals go to a detector and it can be able to see your tissues. So that's quantum reception in real life. But at least here you're seeing a huge magnet where you're going. But that doesn't matter. A 1/100th magnetic field, 1/100th of a refrigerator magnet is enough to actually go and maintain a connection with a cryptochrome receptor in the bird's brain. That's what is being proven. Okay. So now let's step back a little and try to kind of gauge as to what's going on here. One of my favorite books I ever read is what is called The Short History of Nearly Everything, written by Bill Bryson. Bill Bryson is not a scientist. He's a writer who writes travel books. But then one day he said, let me take this travel across history, right? This is a recommended book for anybody who loves science and who wants to get into science. A Short History of Nearly Everything. A pun on the brief history of time of Stephen Hawking, I would say. In that, he asks the first question. He says, who are you, right? And the answer he gives is, you are an intricate arrangement of trillions of atoms. So it's almost like I'm standing you there and I take a pair of tweezers and I take off every atom and just put it like a heap right next to you, right? Once I take a tweezer and take off an atom, that atom doesn't have any mind now. So basically you are an intricate arrangement of mindless particles. But hear this, you're special because this intricate arrangement, which is you, will only exist once. 650,000 hours, that's it. After that, what happens? Those atoms just disassemble and go and assemble somewhere else. Right? But let's get from atoms to the elements. You are just a special assembly of common drugstore chemicals. Carbon, hydrogen, oxygen, phosphorus, a little calcium, a dash of sulfur, and a light dusting of other ordinary elements. Think of it that way, right? If somebody made a bill of fare, like how much does it take to make a human being? Not much, because it's just a common drugstore chemicals assembled together, right? Now, of the billions of species which have been living right now, that have existed since the dawn of time, 99.99% are no longer around. They have been devoured by nature. You and your kind have been around for 3.8 billion years, right? Since the universe came around 13.7 billion years ago, right? So that's what is going on here. You are special, and now I want to actually talk about amongst those common drugstore elements. It is just hydrogen that is important, and in that hydrogen, that one electron is all it takes to get into this quantum world. Now let me give you another great example. Let me just throw out three elements at you. One is called gallium, right? Gallium is, I don't know what it is. I heard it for the first time, right? That's kind of an element, gallium. Arsenic is a thing that's found in pesticides and poisons. It looks beautiful. It's a pink element, right? And then you have manganese. Now when you think about manganese, think about the shine in your cutlery. The spoons and forks, they shine because you put manganese in them. So you got this unknown element, an arsenic which is a poison, and then something which makes your cutlery shine. These are three elements lying at various places in the periodic table. But if you combine all these three, you suddenly come up with this thing called a magnetic semiconductor. That is what is present in your computers, where you hide all your files, your PowerPoints, your words, and whatever. What's going on here? Three different elements which not even related to each other, but when you combine them, you come up with a magnetic semiconductor. Something happens at that basic world of protons and electrons, that when they entangle with each other, you come up with something completely mind-boggling, right? We can't live without these magnetic semiconductors. Now, let's kind of get on to what an atom was when it was first predicted by Rutherford, his planetary atomic model, right? So inside you have the positive charge called nucleus. The word came from the Greek word nux, which simply means a nut. Rutherford called them protons because protons means first. So the number of protons is what determines you, like an element. But those things which revolve around, those are the meat of the matter, the electrons. They are going around in nucleus in circular or elliptical orbits. But what is it that they do, right? When Niels Bohr and Max Planck finally came up with, right, that final atomic model, they told that electrons travel around nucleus in fixed orbits. Fixed has a technical word which is called quantized, right? Quantized is what you call these fixed energy packets. So the energy of the electron is fixed in discrete energy packets, that is called the quanta, right? So the energy is not lost by the electrons while in orbit, but it is emitted out as light, right? So whenever this electron goes from the fixed orbit or the quantized orbit, it's always moving. Whenever it moves, it has to fall back to its original orbit, and when that happens, something comes out, and that is what is called light. This is in essence everything, the governing dynamics of the body. So what is fixed? Quantum. Quantum means how great or how much. What is it that it is gaining? What is it that it is losing? It goes up and down and it emits light, right? That is what life is all about. So a quantum which is always in movement. So that's where the word quantum mechanics comes, right? Now, we went very, very weird from this. They went down to see what this universe is made up of. It's not about protons, neutrons, and electrons anymore. They are actually dug deeper and they came up with basic particles. It's almost like I took a number and I start dividing 1 into half, half into 1/4, 1/4 into 1/8, and so on. Imagine somebody doing that with mathematically on paper, okay, driving down to the basic stuff of universe. And this is a table which they came up with. Enrico Fermi, Satyendranath Bose, and Peter Higgs, they came up with these things, right? So according to Fermi, there are these things called fermions. According to Bose, there are these things called bosons. And of course, finally came Peter Higgs. So look at that. So there is the basic particle called up, U, which makes everything go up, maybe. There is something which makes everything go down, so he came up with a particle called down. And then there is a particle which gives charm. Then there is a particle which gives strangeness, kind of property. Then there is something which makes everything stay at top and something which makes everything stay at bottom. These basic particles now, one of the things that they couldn't divide much is the electron. That's what I'm trying to show to you here. Everything has been split except the electron. Now for every negative electron, they found that there is a electron neutrino, which kind of negates that negative charge. Then you have a muon and a muon neutrino. Then there is a tau and a tau neutrino. Don't even break your head as to how they arrived at these particles. It's all on paper. But once it was on paper, mathematically proven, they actually started doing experiments. And that's what they do at that Large Hadron Collider, as they call it in Europe, right? Now, fermions were all there. Bose came in and said that you could now have a glue to stick these particles. So he came up with gluon. And then there is, everything is about light, okay? Our scriptures, whether you are a Christian or a Hindu or a Buddhist, they say everything is light. Now, what is this light now? In an atomic structure, when an electron goes from that fixed orbit, from one orbit to the other, and it falls back, it emits light. So that's that packet of light or energy which comes out when an electron is moving up and down, that is what is called a photon. And then came these Z and W bosons. These four are called gauge bosons. What they do is, they will take one up particle, say for example, and combine it with two down particles, and lo and behold, you have a neutron. Then you take two up particles and combine it with one down particle, you come up with what is called a proton. So everything in this universe is made by these particles. And then you have some protons, the combinations which are called stout and which become heavy. But in comes Peter Higgs and he said, for those particles to stay together, you need what is a particle called Higgs boson, because he kind of named it after himself and Bose. Higgs boson is what combines these particles so that they have a mass. And once they have a mass, in comes Isaac Newton, who says mass times acceleration, which is the speed, is the force. And Einstein said that if you take that mass and propel it twice the speed of the greatest speedster in the world, which is light, you get energy. So there you go. That's where these particles can go. Mass, acceleration. Everything in your life is about these particles. That's it. Elementary particles in a standard model. And that's what led to people now harnessing these particles. One of the greatest inventions, which is the electron microscope. What is this electron microscope? What is this electron microscope? It's nothing but you take a stream of electrons, which is, say the electrons are something which come out of your eye when you see something, right? So they made an electron gun and they made those electrons go in a straight line. And if you take a collection of electrons and when you hit a sample, whether it's a transmission electron microscope or a scanning electron microscope, it's a beautiful thing. When I work with an electron microscope, we take a sample and you coat it with gold particles, and then when the electron stream hits that gold particle, it sees everything at nanometer precision. Because electron is so small, when it goes and hits something, it actually can decipher that pixel down to a nanometer. That's where you got the virus photograph. So if you go into that world called the quantum world, you are actually looking at the inner space. When a telescope looks at outer space, you are quantum world gives you the intricacies of inner space, and that is much more fascinating than the outer space, right? In comes Reverend Mendel, an abbot. What did he do for us? When the atomic scientists were looking at particles, right? Protons, neutrons, electrons. Mendel, who had a degree in physics, botany, and mathematics, all he had was a pair of scissors in his pocket, some paint brushes, and that photograph which I'm showing you there is his backyard. The backyard has that small vegetable patch. His followers still are maintaining that patch. You see those pea plants there, pink and white, which he crossed and to get those pink flowers, and then they kind of give their progeny. That's Mendelian genetics right there still. That's the backyard of his church. Beyond that fence, you see that marble statue of Mendel. So a pair of scissors, paint brushes, pea plants, a small vegetable patch in his backyard, and he brought about genetics. I don't think there is any other example of any personality in the world who, with just those little bitty things in his pocket and a small garden patch, would have set forth a whole subject, right? But what Mendel gave us was the particle units of life, and that's what he did. He was one of the first interdisciplinary scientists who started getting scientific disciplines into one. What did he trigger? He triggered Erwin Schrödinger. Erwin Schrödinger, when I see that cat going around our science circle now, Schrödinger had this greatest example of a cat, right, in a box, right? He says that there is a cat in a box. You don't know whether it's dead or alive till you open the box. The Schrödinger's cat, right? That's where Erwin Schrödinger. But the same Erwin Schrödinger in the 40s at Trinity College, Dublin, he gave a series of lectures. The first time a physicist was speculating on life. He actually read a book called Mind from Matter by a physicist who became a biologist, Max Delbruck. And he wrote a book called What is Life? The ideas in his book was influenced by Max Delbruck. For the first time, a physicist was looking at life in terms of unit particles. And he read genetics, and he said that gene is a aperiodic crystal. First time. Instead of looking at vitality and all those processes and metabolic pathways, let's now cut down biology to basic units. So he said gene is an aperiodic crystal, an information molecule with codes, just like a Morse code of dots and dashes. Right? Yeah, as Phil is saying here, too bad that he did not discover Mendel's work a century later, right? But for what mattered, it triggered this. A physicist looking at what is life, the simple question he had. This book was read by 3 more physicists, or would I say 2 physicists and 1 chemist. There you have Francis Crick, who was a physicist, Maurice Wilkins, who was a physicist, Linus Pauling, the father of chemical bonding, you could say he's a physical chemist, and 1 bird watcher, an ornithologist, a guy who wanted to become an ornithologist, took a course in genetics, and then he joined these other 3 in what we saw was the race of life. Someday in one lecture, I would love to tell the story of these guys, the DNA story, but that's for another day. But these are the 4 people who read this book, What is Life, which is a 90-page book in print. Required reading for everybody. What an amazing book. It never gets dated. Where a physicist completely dwindles down all the biological principles as units of life, right? That's what got us the DNA revolution. So this is what happened in the quantum paradigm. We dwindled down life to electrons, and these electrons can interact with each other trillions of miles away. Spooky connection over spooky distances, right? Now let's kind of get back to our present. Think about animals which were there, gigantic animals, which again they completely got extinct. They got extinct, right? So there is an animal called T-Rex, which is there is an animal called T-Rex which got extinct, right? It used to stand 5 meters tall. It used to stand 5 meters tall. What kept this animal intact, even at this gigantic height, right? Muscles and muscles of mass. There is this muscle called, there is this biological uh, chemical called collagen, which actually keeps the T-Rex intact. Now, if you go back to Cretaceous time, that's when where the T-Rex, and that's a big misnomer, right? That movie name should have been Cretaceous Park, but maybe it didn't sound attractive, so they said Jurassic Park. But actually, technically, it should have been Cretaceous Park. I don't know how good it sounds. It sounds good to me, but I don't know why they named it Jurassic Park. But that's beyond the matter. So T-Rex was walking. Imagine the T-Rex walking, and suddenly it reaches a riverbed. For all that matters, that collagen which kept its muscles intact was not good enough for it to just stand upright. So it got as if it got into a quicksand. It completely went into that river mud, all going down, and then came the meteors and everything got extinct. But this T-Rex was preserved in that wet riverbed. That collagen became a bane, a curse for it, because it never could swim out. It got crushed into those riverbeds, and one of the main causes. Well, later on came another creature, Homo erectus, which survived all those extinction processes. It became Homo sapiens. Yeah, one of those Homo sapiens, Dr. Mary Schweitzer, one day, she got a fossil bone, one of the first fossils of T-Rex. And when she just put that in an acid, what she thought was fossils are nothing but muds and rocks and minerals just getting into those bones, and if you put them in an acid, they will just disintegrate into something else. She found that when she took out that mass which she put in an acid, it still was sticky. It was almost like a mold. It was like she could stretch it, and to her shock, she found that the collagen was intact. What she saw there was not a fossil, but actual dinosaur meat in that fossil. What's going on? How come this collagen was so beautifully preserved, right? Everybody said, that might be a misnomer. It's not muscle, it's just some sticky glue. So what did she do? She put that enzyme called collagenase, which would break down, and to her amazing sort of a eureka moment, that collagen actually gave the collagenase, when she put it on that collagen mass of that T-Rex, it actually gave her all the products, intact products of some peptides and some amino acids, which are very, very beautifully. So it actually preserved, that collagen was preserved. What does this mean? What is this collagenase doing? How come it didn't act, right? So that's when quantum mechanics came again. Collagenase comes under these things called enzymes, which we all have. Enzymes, as we know, are what are called biocatalysts. They take part in the reaction, but they don't actually change, right? That's what an enzyme is. Quantum mechanics came to explain that collagenase, they took that as a model. Say, for example, there is a reaction going on in your body. Look at those three sets now, which are looking at hourglasses, right? On one side you have the reactants, and the other side you have products. The thing to go from reactants to products is so narrow that somebody has to tilt that hourglass. But that's not the case always. You don't need that tilting, as in you don't need that energy to go from reactants to products. What an enzyme does is, in this particular scenario, it simply stretches that hourglass so that the reactants go to products. It's actually bringing that energy down, as if somebody got that hourglass and stretched it off. Right? Now let's maintain that kind of a paradigm to see where quantum mechanics comes in. It not just stretches the hourglass, but it does something else. Now in the left hand side of the slide, you see the collagen. Collagen is nothing but a peptide, right? It's a protein. So you see those CONH, that is what is known as the peptide bond. That's how all the amino acids in collagen are joined together. When collagenase has to cut that peptide bond, when it has to break it down, it has to go there and it uses water. So water comes in like a biochemical scissors and it snips that CONH bond. So that's what that reaction is. You have a stretch of all these peptide bonds, water comes in and it steals an electron from carbon, right? And then you have another thing, if you look on top, that COO is from an amino acid called glutamate. That glutamate donates an oxygen, and that is what is known as the hydrolytic scissor. It cuts that CONH. But what preserved that collagen in that dinosaur? How come that didn't happen? It's been lying there in that mud, because when this collagenase has to act, it doesn't act alone. That enzyme is not alone. If you look at the down part of the slide, you see there is a teeth-like structure. We call it the molecular incisor, like the incisor teeth which you have. And on top of that, you see that small sphere with a positive sign. That is actually a zinc element. What that zinc element does is, when the water is coming down to cut that CONH, it actually holds on to that proton in the water. So unless or until that zinc gives its permission to cut it, that's why I called it a molecular incisor, and then it has to act in tandem with the glutamate on top. Unless or until these three atoms at the electron level do not coordinate, collagenase cannot act. So all these years in the T-Rex, in that collagen, which was finally cut by Dr. Mary Schweitzer, a Homo sapien who came after all the dinosaurs went extinct, the hourglass was stretched, but what's happening is what is called a quantum tunneling. Quantum tunneling is an additional element which will allow those electrons to not roll immediately into the product side, but at that small stage, it's a very programmed but a beautiful entanglement that happens, which makes all the process so precise. So that's what is quantum tunneling, and that's exactly what happens when those radio waves are coming across walls, across the buildings into your cell phone. So at that stage, it doesn't matter whether your radio waves are crossing planets, interplanetary, but there is a connection between those elements when they go through those things. And Einstein never liked this. He said these spooky distances doesn't happen. But he was the guy who brought about the phenomenon that light comes out in packets called quanta, the photons. And that's exactly what gave him the Nobel Prize. Today we see televisions and DVDs and compact discs and whatnot. That is what is called the photoelectric effect. You're walking into a store and suddenly the doors open, right? That's photoelectric effect. That gave him the Nobel Prize, but still he couldn't believe that at that particular atomic stage, things can interact with each other, right? Now let's come down to something which we cannot live without, which is oxygen. Where is this oxygen coming from? The oxygen comes from this equation. Around us we have carbon dioxide and lots of water. This is the only planet which has liquid water. So every time 6 molecules of carbon dioxide, they combine with 6 molecules of water. When light comes into the picture. So again, light, right? So 6 molecules of carbon dioxide, 6 molecules of water in the presence of light gives you a molecule of glucose C6H12O6 and 6 molecules of oxygen. Oh my god, a play of sixes. Okay, sometimes when I read horror fiction, I say 666 is the number of the devil for what may happen, but the devil in works is right here. 6CO2, 6H2O, C6H12O6, 6O2. This is what is governing life all through the planet. But for this to happen, you need a planet where there is abundant carbon dioxide, abundant liquid water. When we found that there are these green guys everywhere, the plants, right? One photon. Mark, just let's look at this now. One photon. When one photon enters your eye, it just goes and you see something and that's done. Did its job. Makes you appreciate the wonders of the world. But what happens when one photon of light gets into a plant? It makes 16,000 tons of glucose. One photon, one light harvesting complex. That's what is present in the leaves. If one proton goes and hits that one molecule of that light harvesting complex, it is enough to make 16,000 tons of glucose. How the hell is this happening? What's going on? When scientists dug deeper, right? When scientists dug deeper, they found that there were these green bacteria which used to live long time back, even before plants were a thing, because everything in the plants is about chlorophyll, right? So it's about that one photon of light which is coming at a speed of 186,000 miles per second, mind you. It's like, imagine standing in front of a baseball which is hitting you with that speed, what would happen? But these plants are fine. They are capturing that photon of light and making 16,000 tons of organic matter. Where is this one photon going? It's going into what is called the stomata of a leaf. That stomata of a leaf has these pores called porins, which are 1/1,000,000th of a meter. Things get so small. And then it gets into 1 nanometer, a 100,000th of a millimeter, into these stacks inside the leaf called thylakoids. Thylakoids, if you magnify a leaf, you can see these stacks of discs. If you cut open them, you see that green thing called chlorophyll. Inside the leaf, you have trillions of photosynthetic machines. And these things, these stacks are chromophore antennae plates. And the molecule inside is called chlorophyll, which is the second most important molecule after DNA. That's what scientists say. Second most important molecule after DNA. What does it have? Look at the top right hand side. It has this thing called an FMO protein. Of course, in the plants now, it's a derivative of FMO protein, but in that first green bacteria which had chlorophyll, it was called the Fena-Matthews-Olsen protein. Right? What did this FMO do? This FMO, right? Of course, Stephen is telling about development of photosynthesis. First, it was like a purple pigment which kind of evolved into the green pigment here. But it's all about chromophores, like which are attracting light. These chromophore antennae plates have the second most important molecule of DNA. All it is a pentagonal spheres which house these magnesium ions. So every time a photon hits that magnesium ion, it makes a pair. It steals an electron. So there is this particle. There is a particle which is for a brief moment, it's a positive and negative combination. Right in that reaction center, you get this pair of particles. They call it the exciton. If chlorophyll was not there, if there was no pentagonal sphere, no thylakoids, nothing, that photon would just be going randomly, which the physicists would call a drunken walk. but this exciton in combination with FMO, right, that transports into the reaction thing, and now we have what is called the quantum walk. And now, once this quantum walk, meaning a stream of excitons, imagine an army of excitons, one photon hitting that reaction center, creating this quantum walk, it helps this protein called rubisco inside the leaf. Now, rubisco is the most abundant protein on the planet, the most abundant protein. So what it did was, that photon of light would have just gone on a random walk and did nothing else. This exciton, this quantum walk, is what is creating what is called a quantum coherence. A coherence which rivals the best of solar cells. And then what happens? These electrons are captured by a chemical called NADP, which is an electron carrier, and aided by this other ATP, another energy currency, with lots and lots of phosphate molecules, will now capture the air which would have killed us, carbon dioxide. It captures that carbon dioxide and makes this molecule of glucose. And on the side, they make oxygen. So this quantum walk seeded life. That's what I'm trying to say here. Right? Yeah, Stephen just says that rubisco is the most abundant protein. The I version of rubisco is rhodopsin. So imagine the same thing. The photon of light which is entering our eye is being captured by rhodopsin. This over here is what is being captured by the chlorophyll. So I tell my students sometimes, carbon dioxide and water, the crap that comes out of your nostril and our waste, right? Plants are capturing the crap and making us food to eat, C6H12O6, right? And air to breathe. But everything is happening because that photon, that sun, which is sending us those photons, is creating this amazing quantum walk, or the quantum beat, sometimes it's called, which is responsible for the air we breathe. Now let's look at another fascinating phenomenon. When you smell a rose, right, you have these receptor cells in your olfactory membrane which kind of get those odor molecules, and they get into the olfactory nerve, and where you have this broom called the olfactory receptor cells which are activated. They send signals to your brain and you get a particular smell. So the odorant molecule is actually getting into a quantum mechanical phenomena also. What is it doing? It goes and sits in a olfactory receptor. But that's all we knew before. But what we came to realize now is, as soon as it sits in that olfactory receptor, there is a particular proteins called the G proteins. Okay. Now, kind of let's get back in time. When Homo erectus, our ancestor, he lifted himself up on his two legs, he also lifted two things up which we take for granted. One is our eye and the other is the nose. So even the eye and nose was lifted off the ground. That's why our human snout became smaller, the nostrils narrowed, and the mutations are what caused those olfactory receptors. Now this olfactory epithelium is all of 3 square centimeters. That's about it. See how things dwindles down to small universes, like the olfactory 3 square centimeters. It has olfactory neurons, some cilia, brooms, a bulb. The all important compounds are called terpenes, which come from biological entities called plants and flowers. You have a G protein right sitting at that olfactory receptor. It's tethered right there. What happens is, as soon as an odorant molecule is fired, it kind of hits that G protein. Now, if that odorant is not something which we want, it's not pleasant, what happens is that thing is tethered, but it has to get to an acceptor site. It has to bind to a right vibrational frequency. So what is going on? Again, an electron can pop from the donor to acceptor via what is called the quantum tunneling. Once again, quantum tunneling causes a torpedo of G proteins to get to the neuron and send signals to the brain. Now, why is this important? Our first human cognitive behaviors were related to sight and smell. And but for this quantum tunneling, right, when you go after, say, smell a rose, there are many things that the rose gives, a damoxinone or a citronellol, a lot of things, but the one pleasant thing is called the geraniol. That thing has to go and sit on that odorant olfactory receptor complex, and that has to give that stream of electrons which gives you the pleasantness. So even the smell of a rose is governed by quantum mechanics. But if you extend that, you see that it's not just about smelling a rose, but it's more to do with, cognitive behaviors in life. So that was seeded by quantum mechanics. Now let's get back to our Robin for a minute. Before that, is magnetoreception a thing? Monarch butterflies and fruit flies. That's what they looked at. They go like monarch butterflies when they start flying in the autumn. A team of scientists, what they did was they stuck these adhesive tapes on their wings. The adhesive tapes which are so hard to get off your glassware, right, when you buy something in the store. Something related to that, a derivative of that was stuck on those monarch butterflies. They tracked them from Toronto to Texas and beyond. Finally, they found that all the monarch butterflies, they fly in unison from Toronto University down to the Sierra Madre Mountains in Mexico, which was 2,000 miles away. That label said sent to Zoology University of Toronto. What they found was in the antennae of that butterfly, there was, apart from the hypothalamus gland in the brain, the butterfly had a sun compass. The compass looks at the height of the sun, which can vary with longitude and latitude. And when they were digging deeper into the monarch butterfly apparatus, they looked at fruit flies. Fruit flies were Drosophila, the greatest model of biology given to us by Thomas Hunt Morgan. What they found was inside the fruit fly's eye, there is a cryptochrome. There is a pigment molecule, and what was the pigment molecule? The Ph.D, which we saw in the Robin, our friend Robin. That flavin adenine dinucleotide absorbs blue light again, just like it did in the bird, knocks out an electron, its signal travels to the brain. So when they did a mutant, they knocked off this Ph.D molecule, that property was lost. Now look at this. They took that Ph.D gene and they put it in a butterfly. They took that Ph.D gene from a monarch butterfly and put it in the fruit fly mutant, and the magnetoreception was restored. So the cryptochrome that is found in the butterfly could negate a mutant of a fruit fly. They did something, another fascinating thing. They wanted to see the exact location of this compass, and what they did was they saw that the antennae of the monarch butterfly was the thing. So they blackened it. They painted those antennae with black paint, and they found that they could not receive the sunlight. But they took those antennae genes, put it in a fruit fly, and they restored the mutant. This cryptochrome with the FAD gene, was attached to what is called a photolyase domain, and it could actually give the inclination for that monarch butterfly to travel 2,000 miles from Toronto to Mexico. And again, the quantum entanglement of the Earth's magnetic field. Look at how weak that butterfly is in terms of weight or whatever, but it goes exactly to the Sierra Madre Mountains. That quantum entanglement is what scared even a scientist called Einstein. He said, I don't believe this. I don't just believe this spooky entanglement. Where do we go with this? We went back to the Robin now. Let's look at the Robin Odyssey. They put these very lightweight GPS components onto the Robin. And then they created what is called the Emblem funnel. What this Emblem funnel does is, it has like an ink pad. It's a funnel with a mesh. That mesh has a magnetic field which can be controlled. So they can put a Robin inside that funnel, and they can control the magnetic field to see how the Robin flies inside the funnel. And also, at the base of the funnel is an ink pad. So the Robin would sit on that ink pad and start flying, and its footprints will be all over the funnel. So they can track, so they can stimulate a long distance flight and a short flight. And what they did was, they did many interesting experiments. They took the robins which would fly in winter and in autumn and they saw different flight patterns. And when they varied the magnetic field, they could see that the robin flights are beautiful patterned flights inside the funnel. They dissected a robin before and after it took a flight, and I can see on the far lower end of the screen the fat reserves inside when it fed itself lots and lots of insects, right? These fat reserves, these fatty acid reserves, they could even figure out the what you call the biochemical pathways. These fatty acids join to what is called the PPAR molecules, peroxisome proliferator activated receptors. That's what they are called. Peroxisomes are what will take up all your fatty acid food and put themselves, and they are like reserves which will be used later. These fatty acid, basically PPAR is like, it once fat gets into your body, it maintains the homeostasis of the fat metabolism, right? But this, once the fatty acids join to PPAR, they go and interact with circadian clock molecules. One is called the BMAL. It's called the brain and muscle nuclear transporter like molecule. The other is clock. It's a very interesting name, CLOCK. It's actually circadian locomotor output circadian kaput, something like that. I just come up with this name so that they can get a nice paradigm, I guess. But clock and BMAL will now interact with PER, a gene which, the period gene, which regulates the timing of the circadian clock, and of course the cryptochrome. That is where they dissected out the entire apparatus in the Robin Odyssey, and with controlling magnetic fields, they can now see that the pattern is beautifully amazing to see, right? So where does it take us? Schrödinger asked a question, what is life? And he said, gene is an aperiodic crystal. Now we know what that aperiodic crystal is. It's nothing but the DNA. An aperiodic crystal simply means that a molecule with a particular pattern with repeating units, which is a crystal, right? So we have these repeating units called ATGC bonds. ATGC bonds. That's a pattern. It's a beautiful repeat. But why does it make it aperiodic? As you can see, A and T and G and C are always joined by the weakest bonds. What are those bonds? Hydrogen bonds. Oh my God. Coming all together. And now we see hydrogen becomes very important here. What does this hydrogen do? This hydrogen either stays with a nitrogen or jumps off a nitrogen. That's how it is. So when there is an AT base pair, right? The jumping of that electron from nitrogen to hydrogen and back sometimes might cause what is called a tautomeric shift. So A and T may not be the only things that can pair. Sometimes an A can pair with G or a T can pair with C, but that is not happening. That is not happening. Why? Because again, quantum mechanics come into play. We look at what are called quantum leaps. Imagine that hydrogen bond is like a spring. Okay, you see it on the lower left hand side of the table. One of the spring is stretched, the other spring is small. It almost seems like that hydrogen bond that spring can go either ways, but the proton or the hydrogen is in what is called a proton well. One of the wells is deeper, the other well is not so deep. The deeper is the most preferred spot for the hydrogen to be. So the AT is what is maintained, not an AG or a CT pair, though that probability is there, right? So amazing to see that what Schrödinger envisaged is actually happening in real life. So again, the governing dynamics of life is quantum mechanics. Now let's see something else. We delved into magnetoreceptions. So somebody said, could these spooky entanglements, are they telepathic? Are they sending signals out there, right? Is there a quantum world out there? Because your mind is a field, an electric field. Is there a mind that exists in a parallel universe? Because just now we saw how in the DNA itself there are two worlds that can exist. One is what is called the ideal ATGC world, but that proton shifts can cause another kind of DNA. Can you create a universe by perceiving it? Right? But of course, your TV does not create the program. Your TV doesn't create the program. Your TV receives a signal which you see as a program. So your brain cannot create consciousness, but these spooky interplanetary, intergalactic connections between things like a magnetoreception, is something like that happening? Is that something which is more fascinating than what we think, right? So what exactly is the mind, right? Mind is nothing but there you have a sympathetic and a parasympathetic system, where you have the muscles, the myosin, right? That is what is doing all the dog's work. You lift weights or whatever, you feel the pain, but the signals are what go into your brain. What exactly are those signals? Those signals are nothing but a stream of sodium ions. They go through what are called gated channels. So again, when you come down to the molecular atomic world, your brain is nothing but a series of electric signals that are precisely controlled by biochemical reactions. But how far can we stretch this? Look at the right hand side of it, of this slide. You come across these small units where you could be, the spin now becomes very clear here, right? The spin would be like one of them would be in the up direction, the other in the down direction. Just think of two electrons like that. But that's a very ideal world. One is spinning up, one is spinning down. Say for example, one of the electron tilts to a particular direction, right? So if it is ideal, the one direction will be 0 and the other will be 1, right? That's what the binary thing to make a standard computing algorithm. But what this quantum entanglement does is, right, it brings into 3 different kinds of, imagine those 3 things. That unit is what is called as a qubit. That qubit can ideally exist like up and down, or all down, all up. But what if those threads which are connecting them, the interactions which are going within those qubits, can go haywire? One is like 180 degrees off, the other is like up, the other is down, and so on. But that entanglement still persists. If we can harness those kind of reactions, it's like saying that you can seed your thoughts. Your mind can perceive something and that can be actually made a reality. Again, we are anthropomorphic. We always think everything in terms of what we can do as a human being. But hey, the quantum world need not listen to you. It has a life of its own. So where do we go with this? Where do we go with this? Right? Let's start putting everything together. Right? We have a microbiome around us. That microbiome has its own life. It's a compromised world. Everything comes from there. Let's say you can sense, your quantum world can sense that microbiome, and that's what is responsible for every cell inside your body signaling some way, right? Hey, you drink milk and there is an E. coli helping you to digest that. If you don't have that, you have lactose intolerance, right? Now that is what was the world we were living in. We, the microbes, happily living ever after. We have a photosynthesis that is, but now let's look at how that photosynthesis is happening. That photosynthesis is happening because of a quantum beat. We have things like magnetoreceptions that are happening. We are worried about our vision, but look at that small bitty robin, right? These quantum mechanical processes are involved in mutations and repairs inside your body. Look at how that nostril was modified so that you can perceive smell. That smell will influence your cognitive behaviors, your thought processes, flight or fight responses, whatnot, right? Then comes respiration. Where are you getting that oxygen from? From photosynthesis. But without that quantum beat, where would you get that oxygen from? And that will further regulate something in your mitochondria. Mitochondria was once upon an independent bacterium which came into you. Quantum mechanics must have played a big role in that. So we can now look at tumors and diseases. We can look at how populations communicate with each other. We can look at polluting agents, but we get down to the quantum level to see what are those spooky processes, right? We have 34 trillion cells in our body. 34 trillion cells. Every one of those 34 trillion cells has a population of microbes. Another trillion microbes. So you're not alone. Like philosophically, you might say, oh, I'm lonely. No, sir, you're not lonely. You are governed by a microbiome. And bring quantum mechanics into that. Oh my God. No wonder Reverend Darwin, I always say Reverend Darwin because his basic degree was in theology, not biology. That's a big irony, right? So Reverend Darwin once wrote a letter to Joseph Hooker, and he said, it is often said that all the conditions for the first production of living organism are now present, which could ever have been present. Look at that beautiful sentence. But if, and he writes in parenthesis, and oh, what a big if. We could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, and sea. I'm just preserving that English which Darwin used to write, and sea. Present that a protein compound was chemically formed, ready to undergo still more complex changes. At such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed. He was thinking about that spark of life. Man, if he had met Schrödinger, right, what would have been that discussion? But this was what intrigued people. It's not about organisms. It's not about even elements. It's not about compounds, not about chemistry, but quantum mechanics. Quantum mechanics. The origins were not of processes, but the origins of order. Were there patterns in the universe which were governed by these amazing things? No wonder Einstein used to say, God does not play dice. The Lord is subtle but not malicious. Niels Bohr got so irritated, right? When you get irritated, you call a person by his last name without having any pre-additives like doctor or sir. He said, Einstein, stop telling God what to do, right? And he also said, you won your Nobel Prize for photoelectric effect. How can you not think about God not playing dice or he's being subtle, not malicious, right? Yeah, he was such a bore. But then Stephen Hawking put everything in perspective. He said Einstein was wrong when he said God does not play dice. Considering looking at black holes, not only does God play dice, but he sometimes confuses us by throwing them where they can't be seen. That's what he said. But then, but then, Carl Sagan wrote a beautiful book called Pale Blue Dot. Look at that. When we sent out this satellite camera called Voyager, right, in 1977, on Valentine's Day of 1995, if I'm not mistaken. On Valentine's Day, I remember that, February 14th, it took a picture. It just turned itself around at the orbit of Jupiter and it took a picture. I would call it the best selfie ever, right? The best selfie ever. In that beam of light, the Milky Way, in that orange beam of light which is captured, there was one dot, a pale blue dot. There was never another pale blue dot in the entire universe. That pale blue color, yeah, Feb 14th, 1990. Thank you, Feb. 1990. That pale blue dot is because of the liquid water and the quantum mechanical processes which are happening, which gives that pale blue tinge, because the greatest color that is scattered by liquid water is blue. And it became pale blue because of the atmosphere surrounding that oxygen. That is it. That tells you how much of quantum mechanics is not just a random walk, but so precise, because there is no other pale blue dot. We need to preserve this pale blue dot for all we know. So we need to understand quantum mechanics more. Because the same Einstein said, it's not the fruits of scientific research that elevate a man and enrich his nature, but the urge to understand. So I think we understand life. We continue to understand life at the quantum realm. Okay. Thank you. So if you have any questions, I'll be glad to answer.